Hi students, we have already studied one example for intragenic interactions, that is incomplete dominance. The second type, another example for intragenic interactions, that is a codominance. So it is a pattern of inheritance in which normally there is a simultaneous or joint expression of two alleles together. Simultaneous or joint expression of two alleles together. That is a pattern of inheritance. So codominance is a phenomenon in which both the alleles are expressed in the phenotype of an individual. It is a phenomenon in which both the alleles are expressed in a heterozygote individual. That is called as a codominance. We have a number of examples. One such example we have actually the blood group, ABO blood group. This is one example, ABO blood group in humans. Now we have different types of blood groups what is seen already, the genotypes also. A blood group, B blood group, AB blood group and O blood group. Now a person just observed the presence of three alleles for a gene I. This is an I gene. The I gene exists in three forms. One IA, another one IB, another one IO. These are all the three alleles of the I gene. Yes, study. I don't want to explain more about this one. Suppose a marriage occurs between a homozygous A and a homozygous B. What is studied already, the inheritance of blood groups. Now these are all the parents. We have received in F1 individuals with both A and B genes are present. They are equally expressed to produce a character. So in codominance, the two alleles in the heterozygote express themselves individually. So a new character is formed. In the case of dominance, the F1 is resembling any one of the parents. In the case of incomplete dominance, the F1 resembles in between the parents. But in the case of codominance, the F1 resembles both the parents, having the antigen A and B. So this is one example for codominance. Both the characters are expressed equally. There is no blending. So both are present simultaneously. So co-mixed together. So this is one example, ABO blood group. Another example, in the case of plant. In camellia plant, one plant, what is called? The second one example from plant, camellia. Camellia plant. We have to take with reference to flower color. Flower color. We have two types of flower color. One, the red variety. Another one, that is a white. Now, in the F1 generation, there is no blending. There is no actually intermediate color is formed. And both the colors are expressed equally. The flower is having, suppose you are taking a flower, is having just actually white and then what is called the red petals. So there is an indication that both the alleles are present together in the heterozygote. They are expressing equally. And that is what we have in the case of camellia plant, the flower color red and white. There is no intermediate color formation. The red petals and white petals are present scattered, grouped together to form the corolla of the plant. Another example also given in the book. The third example, sickle cell anemia. Sickle cell anemia. Now in the case of sickle cell anemia, we have a normal person. This is one genotype. Another one heterozygote. See, this is what is called heterozygous condition. The third one, you know that one, sickle cell condition. Now this one dies, you know that one, because of homozygous recessive condition. the person dies. Here too we can see there are three different genotypes. Now in the case of this one, the hemoglobin A and sickle cell hemoglobin are actually expressed equally, and it provides an advantage also to the person to just resist against malaria. We can also use this sickle cell anemia for overdominance. Normally overdominance is observed in the case of immune system genes. When one gene is normally dominant over another, when you are getting the heterozygous individual, it is normally superior over the homozygous individuals, providing protection to those individuals who are carrier. For example, this hemoglobin normal persons, HbA genotypes, they are affected by malaria, whereas the heterozygous individuals are not affected either by malaria or by sickle cell anemia. So this is called overdominance. We can also use the sickle cell anemia for overdominance. Many examples for sickle cell anemia can be used. Okay, this is overdominance. The heterozygous individuals are healthier than what is called homozygous individuals. That is different. Secondly, here as per the book it is given another example what is called sickle cell anemia. So these are the three examples for codominance. Now let us see the demonstration of codominance in plants. The demonstration of codominance in plants was noticed or studied through electrophoresis or chromatography for the seed proteins or flavonoid substance. One can study that is about the codominance in plants through electrophoresis or that is chromatography of seed proteins, analyzing the seed proteins or flavonoid substance. That is nothing but the flavor proteins, flavonoid substances. Through this one can demonstrate that is a codominance in plants. For example, a cross was effected between two that is cotton plants, Gossypium hirsutum and Gossypium sturtianum. So, a cross was effected between two cotton plants, one Gossypium hirsutum, another one Gossypium sturtianum. Gossypium sturtianum. Now, these two plants were crossed, and the F1 hybrid was obtained. F1 hybrid. The F1 hybrid is amphiploid. This is amphiploid. Amphiploid. What is amphiploid? Here, these individuals have diploid number of chromosomes. The diploid number is equal to 4x. That is called amphiploid. So the diploid condition is equal to 4x. That means they have 4 sets of chromosomes. In the case of cotton, we have 26 chromosomes. So n is equal to 30, or we can say x is equal to 30. So the normal diploid individual has 4 sets of chromosomes, 4 into 30. So they have 52 chromosomes. That's about the nature of that F1 hybrid. Now the seed protein in that plant or in that F1 hybrid was analyzed by electrophoresis. The parental varieties show that is different patterns of banding protein or different banding patterns of protein amino acid. So different banding patterns of protein. So in the case of electrophoresis, we are getting what is called the banding pattern like this in a gel plate. This is called the banding pattern. So the two plants show different banding patterns of proteins. These two. In the case of F1 hybrid, there was an additive, that is banding pattern. So, the hybrid shows the presence of two types of proteins which are similar to the two parents. So, the proteins of these two parents were inherited here. So, the hybrid, which is normally ampiploid, is having the presence of the two different types of proteins similar to those proteins present in the case of Gossypium hirsutum, Gossypium certian. Now, the genotype, the heterozygote genotypes produce, actually control a phenotype which is distinctly different from the homozygous genotypes of the parents. So, the heterozygote genotype controls or produces a phenotype distinctly different from, that is, the parental genotypes. When actually these plants are crossed, say an example, in the case of any codomiance, when F1 plants are crossed, we are getting the ratio 1:2:1. As in the case of incomplete dominance, here also we are getting the phenotypic ratio 1:2:1 and also identical genotypic ratio of 1:2:1. For example, in the case of marriage between persons with the A blood group and persons with the B blood group, we are getting just only one type. For example, AB blood group, that is a hybrid. We can see heterozygous. So when AB, they form gametes. So two types of gametes are produced. Two types of gametes. If marriage occurs between two AB blood group persons, so we are getting also here two types of blood groups. So we are getting what is called actually A blood group, then AB blood group, and then B blood group. These are the products obtained when marriage occurred between two individuals which are, that is, heterozygous for A and B. Now the ratio is 1:2:1. So both the phenotypic ratio and the genotypic ratio appear in the case of marriage between AB parents is equal to 1:2:1. So either in the case of incomplete dominance or in the case of codominance, the result will be the same. The phenotypic and the genotypic ratios would be similar, 1:2:1. What is lethal gene? An allele which has the potential to cause the death of the organism is called as a lethal gene. So, the term lethal refers to those changes in the genome of an organism which normally causes, that is, which is enough, severe enough, we can say which is severe enough to cause the death. So the term lethal refers to those changes in the genome of an organism which is normally enough to cause the death of the organism. The effect is enough to cause the death of the organism. We can say that is called actually lethal genes. What is lethality? Lethality is a condition which causes the death of certain genotypes, that is prematurely. Lethality is a condition which causes the death of certain genotypes individually. Now, we have two types of lethality. One is called dominant lethality, another one recessive lethality. Recessive lethality, this is one type of lethality. Another one, dominant lethality. These are the two types of lethality. Now we are going to study first one, what is dominant lethality, the recessive lethality, then go for the dominant lethality. Now we are taking the example in the case of antirhinum species. anti-rhino. You know the snapdragon. We have studied already the flower color, snapdragon, for incomplete dominance. Here we are going to study for dominant lethality. The example could be yellow mice. Yellow mice. That is example. First, let us take. In both the cases, you know, the fully dominant allele or the recessive allele cause or kills the carrier organisms only in homozygous condition, but not in heterozygous condition. I'll repeat once again. The fully dominant allele or the recessive allele, they cause or they kill the carrier organisms only in homozygous condition. So, the phenotypic ratio will be 2:1 or 1:2. These are the two ratios apply. Now, in the case of recessive lethality, the phenotypic ratio is 1:2. In the case of actually that is dominant lethality or yellow mice, the phenotypic ratio is 2:1. I mentioned about fully dominant and fully recessive. Here, for example, fully recessive, I am taking like this. This is fully recessive. This is what is called fully dominant. So here, the phenotypic ratio, all the genes are lethal when they are in homozygous recessive condition. That is called recessive lethality. In recessive lethality, it causes the death of the organism in homozygous recessive condition. In dominant lethality, the genes in dominant homozygous condition cause the death of the organism, accordingly dominant lethality. So the one which produces death in dominant homozygous condition. Here the one which actually in homozygous recessive condition kills the organism. So this is called recessive lethality and dominant lethality. Now let's go for the example of recessive lethality. As students, I want to give explanation for recessive lethality. I mentioned earlier the recessive lethality results due to the recessive genes in homozygous condition and that kills the organism. Hence called recessive lethality. So it was observed by Bohr in anti-random species. A cross that is normally a green and white plants and obtained a heterozygous variety what is called aria. Now the heterozygous is having one dominant gene, one recessive gene. So when these two individuals are self-crossed, male and female, we have received the gametes, here 50% with capital C and 50% with a small c. In both the individuals, they are producing, that is 50% capital C and 50% small c gametes. Now, because of random combination, we have received the F2 generation with the following individuals. One green with the dominant gene, homozygous, and two aria, the heterozygous individuals, and one that is white. But as per the phenotypic ratio in the case of that is Mendelian inheritance, we are getting 3:1 phenotypic ratio. But here, the recessive pair or the individual with the homozygous recessive dies, so that the phenotypic ratio of 3:1 is modified. Now, as it is without chlorophyll, the white parent dies. Now we have the modified phenotypic ratio 1:2. One green and two aria were survived. So the phenotypic ratio is 1:2 and so also genotypic ratio 1:2. That is one capital C homozygous and two aria with heterozygous condition. So this is the explanation for the recessive lethality. Individuals in recessive homozygous condition died. This is because of recessive lethality. The genes normally in recessive condition, homozygous condition, kill the individual because of the lacking of chlorophyll. Now let us discuss about the dominant lethality, that is with reference to the yellow mice. In the case of dominant lethality, now the dominant gene in homozygous condition dies. In the previous one, recessive lethality, the recessive gene in homozygous condition dies. Now, here the yellow mice. Yellow mice was crossed with a non-yellow mice. We are getting a heterozygous yellow mice. That is in the F1 generation. Now we are considering this is the parallel generation. So, two yellow mice which are heterozygous for the color were crossed. They produce the haploid gametes. The haploid gametes. 50% with capital Y and 50% with the small y. That is 50% with the dominant allele and 50% with the recessive allele. Now, by means of random combination, we have received the following results in F1 because we are taking these two are the parents, so the result obtained is F1. Now, in the case of this one, we are getting actually there is one homozygous L and two heterozygous L. These four heterozygous L and one homozygous non-L. Now here, the dominant gene in homozygous condition causes the death of the individual. So this homozygous dominant yellow dies. So this is the individual dies. So we have only two heterozygous yellow and one non-yellow. So the phenotypic ratio would be 2:1, and so also the genotypic ratio 2:1. Two heterozygous yellow and one homozygous non-yellow. So in the case of recessive lethality, we have 1:2 ratio. This is in the case of homozygous recessive lethality. The homozygous recessive individuals die. But in the case of this one, the ratio is 2:1 because the individuals with the dominant allele in homozygous condition die. So this homozygous dominant yellow die. So we are getting only two heterozygous yellow and one non-yellow variety. So anyway, I mentioned already, the fully dominant allele or the fully recessive allele kills the carrier only in homozygous condition. If it is a dominant allele in homozygous condition, it causes the death. If it is actually a recessive allele in homozygous condition, the case of previous case, it causes the death. So we are getting the phenotypic ratio either 2:1 for dominant lethality or 1:2 for recessive lethality. Don't forget, the ratio for dominant lethality is 2:1. The ratio for recessive lethality is 1:2. There are three types of inheritance. Generally, we can say 1. A single gene influences a single trait. This is called monogenic inheritance. One gene, one trait. It's proposed by Mendel, hence called Mendelian inheritance. It's also called qualitative inheritance because the expression depends on the quality of the genes, either dominancy or recessiveness. Now the second one, a single gene controls many traits, and this is called pleiotropy. A single gene controls many traits, this is called pleiotropy. Now the third condition, many genes are involved in controlling a character, and they are called together as polygenes. The inheritance is called polygenic inheritance. So one gene, one trait. One gene, many traits called pleiotropy, and many genes control one trait, it is called polygenic inheritance or multiple factor hypothesis. Now, let us go for pleiotropy. The first one we already studied, the second one now we are studying, the third one we'll talk about later, the same lesson. Now, in the case of pleiotropy, a single gene has effects on multiple traits or characters, altering the phenotype of an organism. That is called pleiotropy. The gene which influences many characters of an organism are called pleiotropic genes. Now it was observed by Mendel while he was actually doing the experiments, the breeding experiments with the garden pea plant. Now he observed in the case of garden pea plant, three characters. One, the seed color, another one, the flower color, the third one, a spot in the axil of the leaf. It took one plant variety, brown seed, another one, the purple flower, another one, dark spot. It was crossed with a variety of pea having light seed, white flower, and no spot in the axil of the leaf. Okay, these three characters are inherited together as a single unit. This is due to the patterns of inheritance in which these three traits are controlled by a single gene with the dominant and recessive alleles. These three characters are actually inherited together with a gene with the dominant and recessive alleles. Even sickle cell anemia also given as an example for this pleiotropy. Once again I wanted to repeat, these three normally characters are inherited together as a single unit. This is due to the pattern of inheritance controlled by a single gene with the dominant and recessive alleles. Even sickle cell anemia is given an example in the book for pleiotropy. Even PKU also an example for phenylketonuria, the disease, an example for pleiotropy.